بسم الله الرحمن الرحيم. Our video today is about a new subject, which is extracellular vesicles. And again, before we start, I'm reminding you to subscribe to the channel and activating the alarming bell if you are interested in getting any new video. Here's my first reference, which is published in Cells 2019, and its title is Overview of the Extracellular Vesicles, Their Origin, Composition, Purpose, and Methods for Exosome Isolation and Analysis. Most of the communication between cells in our bodies occurs through the transfer of bioactive molecules, which include proteins, nucleic acids, metabolites, and lipids. These molecules are transported either through passive diffusion or by carrier molecules such as extracellular vesicles. Extracellular vesicles are lipid-bound vesicles secreted by cells into the extracellular space. And there are three main subtypes of extracellular vesicles, which are microvesicles, exosomes, and apoptotic bodies. Extracellular vesicles content or cargo consists of lipids, nucleic acids, and proteins, specifically proteins associated with the plasma membrane, cytosol, and those involved in lipid metabolism. These bioactive molecules delivered through extracellular vesicles can cause physiological changes in the recipient cells. The contents of extracellular vesicles containing biomolecules vary depending on factors such as tissue of origin, immune set point, and cellular context. The study of extracellular vesicles has greatly contributed to our understanding of cell-to-cell communication and cancer metastasis. Moreover, their potential clinical application as carriers of biomarkers for diagnostic purposes has been demonstrated. The first subtype of extracellular vesicles is exosomes, which are intraluminal vesicles that are contained within a singular outer membrane and are released by various cell types. Exosomes have been detected in several body fluids such as plasma, urine, semen, saliva, bronchial fluid, cerebrospinal fluid, CSF, breast milk, serum, amniotic fluid, synovial fluid, tears, lymph, bile, and gastric acids. Exosomes are created through an endosomal pathway, and typically their size range from 30 to 150 nanometer in size. Exosomal vesicles are produced through the inward budding of the early endosomes, limiting membrane, which eventually matures into multivesicular bodies through this process. Actually, early endosomes, which originates from the inward budding of the cell's plasma membrane, and microvesicular bodies are involved in the endocytic and the trafficking functions of the cell's material. Specifically, they are involved in protein sorting, recycling, storage, transport, and release. In this diagram, we can see the formation of exosomes or the other extracellular vesicles, which are microvesicles. So, as regards exosomes, first we have what's called early endosomes, which is formed from inward budding of the cellular membrane in the process of endocytosis, and these early endosomes are responsible for sorting and trafficking process of engulfed materials or cellular materials, and then these early endosomes will make inward budding of its limiting membrane. Limiting membrane is a membrane which separates the endosomes from the cytoplasm, and then this early endosomes will become multivesicular endosomes by making this inside many vesicles, and these multivesicular endosomes either making let endosomes and fuse with lysosomes to degrade the materials, or to go again to the cell membrane and fuse with it and release its contents, which include exosomes. And the exosomes already have some machinery items from its origin. While the microvesicles is larger than the exosomes, and it's formed from outward budding of the cellular membrane, as we can see here in the diagram. As I said, multivesicular bodies are either sent to the lysosomes to be degraded along with all of its contents, or fused with the cell's plasma membrane to release its contents, including exosomes. The factors that determine the fate of a specific multivesicular bodies are not well understood. Research has shown that the destination of a given multivesicular body is influenced by the amount of cholesterol present within it. So, more specifically, multivesicular bodies that are rich in cholesterol are released through cell membrane, while those with identical morphology but lacking cholesterol are directed towards the lysosome for degradation. The endosomal sorting complex ESCART required for transport pathway is responsible for controlling the formation and release of microvesicular bodies and exosomes. And although the exact mechanisms are not yet fully understood, it's believed that the microvesicular bodies formation can be stimulated by growth factors, and the cell adjusts its production of exosomes based on its requirements. Exosomes initially were considered as a mechanism for cellular waste removal. However, exosomes have now been discovered to play important roles in intercellular communication, cellular homeostasis, and even cancer progression. Also, exosomes have demonstrated their ability to activate immune responses by acting as carriers of antigen-presenting molecules. Exosomes have been shown to facilitate myelin formation, neuronal survival and growth, promoting tissue repair and regeneration within the nervous system. However, in the central nervous system, exosomes can also carry pathogenic proteins, including beta-amyloid peptides, alpha-synuclein, and superoxide dismutase, which may contribute to disease advancement. Exosomal research is frequently focused on investigating their potential as vehicle for disease biomarkers. For example, alpha-synuclein, a protein linked to Parkinson's disease, has been identified in exosomes found in both plasma and cerebrospinal fluid. Also, a recent review has centered on extracellular vesicles as markers for glioblastoma, while urine-derived exosomes have shown promise in reflecting acute kidney injury. Also, markers for pancreatic and lung cancer have been detected in exosomes, indicating success in using exosomes as a potential diagnostic tools for various diseases. So, exosomes are an ideal biomarker carrier, as they are present in body fluids like urine, blood, which enables the adoption of minimally invasive or non-invasive methods like liquid biopsy for diagnosing and monitoring a patient's response to treatment. Actually, exosomes offer another potential application in the clinical setting by enabling the monitoring of a patient's response. Also, some researchers have suggested using exosomes for vaccine development and other immunological applications. The antigen-presenting ability of exosomes presents an important opportunity. Also, exosomes have a lengthy half-life and are well tolerated by the human body. Also, exosomes can cross cellular membranes and potentially target specific cell types, so they are excellent candidates for immunological applications and ideal for drug delivery system development. And although techniques for introducing RNA and protein into exosomes, directing them to a particular region of the body, are still in development, the potential to load protein and genetic material into exosomes represents another advantage that makes them an attractive drug delivery system. Also, research has shown that mesenchymal stem cell exosomes can act as therapeutic agents and aid in reducing tissue injury. The second type of the main subtype of extracellular vesicles is microvesicles. So microvesicles are extracellular vesicles that is formed by direct outward budding or pinching of the cell's plasma membrane, as I said in the previous diagram. The size of the microvesicles typically range from 100 nanometer up to 1 micrometer in diameter. A nanometer is 1,000 times smaller than a micrometer, so it's much bigger than the exosomes. The route of microvesicle formation is not well understood. However, it's thought to require cytoskeleton components such as actin and microtubules, along with molecular motors such as kinesins and myosins, and fusion machinery like what's called SNRs and tethering factors. The production of microvesicles is affected by the physiological condition and microenvironment of the donor cell, while the consumption of microvesicles is affected by the recipient cell physiological state and microenvironment. The absorption of microvesicles appears to be dependent on energy, as lower temperatures have been observed to inhibit uptake by the recipient cells. The clinical applications and purposes of microvesicles are comparable to those of exosomes. The third main subtype from extracellular vesicles that was mentioned in this paper is apoptotic bodies. Apoptotic bodies are released by dying cells into the extracellular space. They are reported to range in size from 50 nanometer to 5,000 nanometer in diameter, with the size of most apoptotic bodies tend to be on the larger size. These apoptotic bodies form by separation of the cell's plasma membrane from the cytoskeleton as a result of increased hydrostatic pressure after the cell contracts. Apoptotic bodies contain intact organelles, chromatin, and a small amount of glycosylated proteins. Here is another nice paper that was published in International Journal of Molecular Science 2023, and its title is Extracellular Vesicles and Viruses to Intertwined Entities. In this paper, they said that extracellular vesicles are derived from multivesicular bodies, exosomes, or from the plasma membrane, microvesicles, or other organelles such as autophagosomes. So in this paper, they substitute the apoptotic bodies by the autophagosomes as the main subtypes of extracellular vesicles. Extracellular vesicles derived from autophagosomes are formed through a non-classical mechanism in which autophagosomes fuse with the endosomes or microvesicular bodies, or alternatively fuse directly with the plasma membrane rather than undergoing fusion with lysosomes. Extracellular vesicles have the ability to initiate immune responses against pathogens and tumors by carrying antigens and immune-stimulating factors. They also aid in maintaining cellular homeostasis by releasing harmful components such as nuclear DNA in the cytoplasm. Moreover, they are involved in various biological processes such as pregnancy, stem cell differentiation, and injury recovery. Extracellular vesicles released by mesenchymal stem cells from adipose tissue play a critical role in regulating cell proliferation, migration, apoptosis, as well as in processes such as angiogenesis, nerve regeneration, and immune responses. Extracellular vesicles have the ability to transport misfolded amyloidogenic peptides linked to neurodegenerative diseases like Parkinsonians and Alzheimer's. Also, they have the ability, they contribute to cancer progression by stimulating cell proliferation, migration, and angiogenesis, or carry apoptotic and inflammatory molecules that can result in cell death and other inflammatory diseases. The cellular machinery used in the production, packaging, and secretion of extracellular vesicles is frequently used also by viruses for their own replication, assembly, and egress. Actually, viruses have the ability to enhance extracellular vesicles production or manipulate extracellular vesicles to disseminate their own genetic material or protein. In contrast, extracellular vesicles can play a significant role in controlling viral infections by transporting immunomodulatory molecules and viral antigens to initiate antiviral immune responses. So, extracellular vesicles produced by viruses have the potential to serve as strong immunomodulatory agents, which are capable of either stimulating or inhibiting the immune system. This may result in the development of chronic inflammatory diseases or facilitate immune evasion and progression of conditions like cancer. So if they stimulate more the immune system, they can result in chronic inflammation, chronic inflammatory disease, or if they suppress more the immune system, they can result in a progression of cancer. However, interactions between viruses and extracellular vesicles can serve as a promising immunotherapy approach for addressing a broad range of disorders. RNA and DNA viruses, regardless of whether they are enveloped or not, utilize the extracellular vesicles pathway to release their viral particles, proteins, and nucleic acids. Also, they utilize this pathway to secrete host factors that help in viral infections. So, extracellular vesicles and viruses serve as essential carriers for a variety of therapies. In the field of oncology, both extracellular vesicles-based therapies and virotherapies are currently under development. So, given the capacity of virus extracellular vesicles to augment viral dissemination and stimulate immune responses, the utilization of a combination of oncolytic viruses and extracellular vesicles has the potential to enhance the effectiveness of cancer treatment. So, encapsulating oncolytic viruses within the extracellular vesicles offers a secure delivery mechanism and improved uptake of the oncolytic viruses to the tumor site, resulting in enhanced efficacy of the virotherapy. Although this approach is still in the developmental stage, it holds a big potential in the area of cancer therapy. Lastly, I hope that you have enjoyed the video, and I'm reminding you again to subscribe to the channel and activating the alarming bell if you are interested in getting any new video, and thank you.